Hello students, we are going to talk about human immunodeficiency virus and which is known to cause AIDS disease. AIDS is called as, the full form of AIDS is autoimmune deficiency syndrome. The HIV disease causing HIV-1 causes AIDS and it was first reported in United States in 1981 in Los Angeles, New York and San Francisco. And then and thereafter it spread as an epidemic. Usually the group of patients which displayed unusual infections including the opportunistic fungal infection, pneumocystis carnia which causes a pneumonia called PCP, they were being identified as immunodeficient persons. And some of the patients has Kaposi's sarcoma which is an extremely rare skin tumor. In case of AIDS patients, the T cells, they carry these CD4 markers and they were being infected. When we talk about the epidemiological background, we see that this disease, it was most prone, or the people who were at high risk of AIDS, they were either homosexual males or they were promiscuous heterosexual individuals which were having either sex and their partners, or they were having the intravenous drug users, or they were persons who received blood or blood products prior to 1985. By December 1998, approximately 33.4 million persons, they were living with AIDS. And most of these were in sub-Saharan Africa, Southeast Asia, North America, Western Europe, and about 80% of those affected were men. Whereas in Africa, the equal number of women and men had AIDS. Now, when I talk about the causative agent, that is a human immunodeficiency virus, this virus, the mode of its transmission was either through homosexual or heterosexual intercourse or through the recipient of blood or blood products, and it was even passed from mother to infant. Before tests for HIV in the blood supply were routinely used, the patients who received blood transfusions and hemophiliacs who received blood products were at higher risk of HIV infection, HIV-1 infection. Now, approximately what happened is, in infected mothers which were treated with antiviral drugs, approximately 30% of the infants born to them, they were prone to the infection. And the possible vehicle of passage from mother to infant infection was a blood transfer during the birth process or milk in the nursing period. Transmission of infected to an uninfected individual is most likely by transmission of HIV infected cells, in particular macrophages, dendritic cells and lymphocytes. 75% of the cases of HIV transmission, they are possible to the heterosexual contact, while the probability of transmission by vaginal intercourse is lower than by any other means, such as intravenous drugs, which are used, and likelihood of infection is greatly enhanced by the presence of sexually transmitted diseases. This was a mode of transmission which this virus followed. If you talk of the HIV virus, it is a retrovirus. It was first discovered and characterized in the laboratories of Luc Montagnier in Paris and Robert Gallo in Bethesda. The retroviruses, their genetic information, it is always in the form of RNA. And they are known to have carry out the reverse transcription, that is from RNA to DNA. And the reverse transcription enzyme has a major role to play, as the reverse transcription enzyme reverses the normal transcription process and it makes a DNA copy of the viral RNA genome. And this copy is called as the provirus, which is integrated into the cell genome and it is replicated along with the cell DNA. When the provirus is expressed to new virion, the cell lysis takes place. The proviruses, they remain latent in the cell until some regulatory signals start the expression process. So see, if the provirus, it is remaining latent, it means some of the infected person, they can act as reservoir hosts without showing any symptoms of infection. The other human retrovirus which is there is human T cell lymphotrophic virus 1 or HTLV1. Human T cell lymphotrophic virus 1, it is again a retrovirus. It is endemic in southern parts of Japan, in the Caribbean. Most of the infected persons with HTLV1, that is human T cell lymphotrophic virus 1, they have no signals of the disease. But a small percentage, they develop very serious illness, either about T cell leukemia, which is aggressive and usually fatal, and which is disables the neurological disorders. It is called as HTLV1 associated myelopathy. Comparison of their genomes that in sequences revealed that HIV1 is not a close relative of HTLV1. However, the similarities in overall characteristics, they led to call the virus of the AIDS as HTLV3 in earlier reports. The related human virus HIV2, which is less pathogenic in humans than HIV1. HIV2 is similar to viruses isolated from monkeys. It infects some of the non-human primates that are not infected by HIV1. Viruses which are related to HIV1, they have been found in non-human primates. If you look at the viruses, the variants which are produced, the variants of SIV virus, that is simian immunodeficiency virus, which causes diseases in certain infected monkeys. Normally, the SIV strains, they cause disease in the normal host but produce immunodeficiency similar to AIDS when injected into another species. Example, the virus from African green monkeys, that is SIV. The SIV virus from African green monkeys, it is present in high percentage of a normal healthy African green monkey, which is wild. And when SIV from the African green monkey is injected into macaws, it causes lethal immunodeficiency. So number of other animal retroviruses may have more or less similar to HIV1 have been reported. This includes the feline and bovine immunodeficiency viruses, the mouse leukemia viruses, so on and so forth. Now looking at the structure of this virus in the slide which I have shown, if you look at the structure of this virus, it's a figure which is being shown the electron micrograph. It is an electron micrograph of HIV virions, which is magnified 200,000 times. You can see in the picture the glycoprotein projections, which are faintly visible as knobs. They are extending from the periphery of each virion, and each virion is expressing 72 glycoproteins projections, which is composed of GP120 and GP41. GP41 is a transmembrane molecule, and it is comprising of the lipid bilayer of the viral envelope, whereas GP120, it is associated with GP41 and it is serving as the viral receptor for CD4 on the host cells. The viral envelope, it derives from the host cells and contains some host cell membrane proteins, including class 1 and class 2 MHC molecules. Within the viral envelope, of the viral, there is a nucleocapsid, which is a nucleus layer of protein, which is called P17, and there is an inner layer of protein which is called as P24. The genome is usually single-stranded RNA, which is associated with two protein molecules, that is reverse transcriptase and a nuclear protein P10, which is a protease. P32 is an integrase protein. Later on in our studies, we will be studying about the role of reverse transcriptase enzymes and the protease enzyme. Next.